Yeah, welcome to the second session of today's research symposium, which will highlight connections between the gut microbiome and the immune system. My name is Alex Carr. I'm a postdoc here at the Institute for Systems Biology in the lab of Dr. Sean Gibbons. And it's my pleasure to introduce our two speakers, Drs. June Round and Melody Zhang. Our first speaker, Dr. June Round, did her PhD at UCLA and her postdoc at Caltech, where she worked with Sarkis Mosmania. She opened her own lab in September 2011 at the University of Utah, where she is the H.A. and Edna Benning Presidential Endowed Chair. Her lab utilizes microbiology, immunology, bioinformatics, and mouse models of disease to elucidate interactions between the immune system and the microbiota, and she seeks to harness the power of the microbiota for therapeutics. And with that, Dr. Round, please take it away.
Thanks. Good morning, everyone. Thanks, Alex. Thanks to the organizers for inviting me. So what a great conference. The first two talks were awesome. I'm really excited to be here today. So my lab's interested in the interaction between the microbiota and the host immune system. And we really look at this relationship as it pertains to promoting health. So instead of looking at this interaction through the lens of pathogenesis, where we're trying to look at the disease state and identify microbes that might be inducing that disease state, we're really interested in microbes that are present during the healthy state and perhaps gone during the disease state, to really identify protective microbes. And we look at this relationship in a variety of different diseases, including IBD, various cancers, and obesity. But the story that I'll share with you today really centers around diabetes. So in thinking about what I was going to talk about, since I'm in a session with Melody Zhang, I decided to talk about our early life microbiota work, but I wanted to just highlight two very recent publications from the lab that have really identified various microbial molecules and their effects on the host. The first is that we've identified a bacterial polysaccharide from B. uniformis that activates NK cells to protect from CRC. And this was recently published, and it's exciting because there have been lots of microbes that have been identified to activate anti-tumor T cell immunity, but this is really one of the first organisms that has a molecule that activates NK cells, but not T cells. And this bacterial polysaccharide is really delivered through outer membrane vesicles from the bacteria. So if you're interested in anti-cancer immunity, please check out that paper. The other paper that just came out this month, and we're actually collaborating with Sid's group on this story, is that we've identified another bacteria called Thericibacter, and how it's able to prevent obesity. And individuals with obesity actually lack this bacteria. And we've shown that this organism is able to suppress obesity by suppressing host ceramides within intestinal epithelial cells. And it does this by secreting a specific lipid. So we're actually working with Sid's group to identify what this lipid is and the genes in Thericibacter that make this lipid. So if you're interested in obesity and the microbiome, check out that paper. But today we'll be talking about a story that involves pancreatic immune interactions. And like most of the work that comes out of my lab, this is really a team science with lots of collaborators. So I just wanted to highlight the collaborators right off the bat. So the first one is Charlie Murtaugh, who is an expert in pancreatic development, and he's at the University of Utah. The other collaborator in this project really stemmed from studies done in Karen Gilliman's lab at University of Oregon, where she worked with at the time, her PhD student, Jennifer Hill, and understanding in zebrafish how the microbiota influence the pancreas. And then also at the University of Utah, a microbial ecologist, Zach Stevens, were all instrumental in getting the story done. And all the work that I'll show you today was performed by Jennifer Hill, who came to my lab as a postdoctoral fellow and has recently moved to the University of Colorado Boulder to start her own laboratory. So the focus of the story today, the cell type we'll be talking the most about, is a beta cell. And before Jennifer came to my lab, I didn't really think about beta cells a lot. But beta cells are housed in the pancreas, which is directly connected to the gut. And these cells are really important for the production of insulin in our bodies and regulation of blood glucose levels. When thinking about diseases that are influenced or that are part of the pancreas, we often think about type 1 diabetes. And type 1 diabetes is a disease where the immune system inappropriately infiltrates the pancreas and specifically targets the beta cells for destruction. And once beta cells are destroyed, they don't readily get replenished because there's a really specific window of time where these beta cells develop. And I'll talk a little bit about this in the coming slides. The primary treatment for type 1 diabetes has been for over 100 years exogenous insulin, which is onerous, expensive, and thus, many people are working on identifying effective cures and or treatments. But when thinking about a treatment for type 1 diabetes, it's really going to involve two specific focuses. And the first one is, of course, blocking that autoimmune response, because that immune response is what's destroying those beta cells. But then, as I had mentioned before, once beta cells are destroyed, it's very difficult to get them replenished. So the second tier of an effective cure is going to be understanding how we can replenish lost beta cells. And the story that I'm going to tell you today really focuses on the second part of a potential therapy for type 1 diabetes. So type 1 diabetes has some genetic components. Indeed, 70% of individuals with type 1 diabetes have a predisposing HLA risk allele, so they have a certain MHC class type. But of all the people that have this predisposing risk allele, really only 3 to 7% of children will go on to develop type 1 diabetes. So the field has really focused on other things that might influence this disease. One of them, of course, being the microbiota. And studies that have come from Ramnick and Xavier's lab have followed the microbiota in individuals that have this predisposing risk allele over their first couple years of life. And they've identified that the microbiota found in individuals with type 1 diabetes compared to those individuals that do not go on to develop type 1 diabetes is really different in their diversity, in that individuals without type 1 diabetes but have the same predisposing risk allele, they have a much higher diversity of organisms, so lots more different types of organisms. So whenever I see something like this, this correlation analysis, this always makes me think that perhaps the disease state is happening because individuals with this disease are lacking specific microbes that are promoting the healthy response. So since we're talking largely about beta cell development, and I know this is an immune session, so I promise you that the immune cells are coming, I wanted to just briefly get everyone on the same page with respect to beta cell development. So early in life, we're all readily familiar that we're born fairly sterile, and then over the first 2 years of life, we assemble a particular microbiome that around the age of 2 to 3 starts to look a lot like an adult microbiome. Similarly, when we look at the development of beta cells, we're born with a certain number of beta cells, but in humans, during the first 6 months of life to a year, these beta cells rapidly divide, and then it sets the beta cell mass that you will have for the rest of your life. Beta cell proliferation around one year of life kind of wanes off, and the number of beta cells that you develop during that time is basically what you're going to have for the rest of your life, unless, of course, something happens. So at this really early window of life where we're assembling the microbiome and also establishing our beta cell mass is really important. So when Jennifer joined my lab, she was interested in understanding whether or not the microbiota influenced beta cell mass in a mammalian model. She had published some beautiful studies in Karen Gilliman's group where she had identified that the microbiota was important for beta cell development in zebrafish, but she really wanted to move to a mammalian model. So she came to my lab and first looked at beta cell mass. And you can just think about this as the insulin producing capacity in your body. So the higher this bar is, the more insulin producing capacity that you have. And she showed in animals that had adult animals that were born and reared in a completely sterile environment, these germ-free mice, that they had significantly less insulin producing capacity. And she did this in a variety of different mouse backgrounds and showed it's not background specific. So this was indicating that indeed the microbiota is important for the development of our insulin producing beta cells. But what she didn't know is that these animals had lived a lifetime with no microbes. So she was interested in this early window of life that I have just described. And wanted to ask questions about whether this is just an accumulation of living a whole life without microbes, or really is it that early life window that's important? So she designed a really nice experiment where she perturbed the microbiota with antibiotics during 3 distinct windows of life. And the antibiotics we used were this, kind of antibiotic bomb, 4 cocktails. It's really good at depleting the microbiota. So she introduced these antibiotics during embryonic day 14 through postnatal day 4. She then withdraws the antibiotics and lets those animals grow up to adulthood and then analyzes beta cell mass. The second window of life that she perturbs the microbiota in was postnatal day 4 to postnatal day 12, again, removing those antibiotics and letting the animals grow up to adulthood. And the final window that we probed was postnatal day 12 to postnatal day 21, again, removing the antibiotics, and the microbiota then rebounds, analyzing those animals at adulthood. So when we go and look at the insulin producing capacity, when we perturb the microbiota during these early life states, we found something pretty interesting. So I've already shown you that a lifelong germ-freeness in these adult animals leads to less insulin producing capacity. If you put animals, SPF animals, on a lifetime of antibiotics, so you start putting them on antibiotics when they're born and analyze them at adulthood, they also have a beta cell mass that looks much like a germ-free animal. But if you perturb their microbiota during this first window or the second window that I described, they actually develop a beta cell mass that looks very similar to a normal, unperturbed SPF animal. So it doesn't seem that perturbation during these first 2 windows of life seem to matter for beta cell development. But interestingly, if we perturbed the microbiota during this short 10-day window between P12 and P21, despite the fact that the animals were born with a normal microbiota and they develop a microbiota after this antibiotic perturbation, their beta cell mass looks very much like a germ-free animal. Moreover, when we go and look at the ability of these animals to clear glucose, we find that their fasting blood glucose levels are significantly higher when they have had neonatal antibiotics. They don't clear blood glucose as well. And consistent with this, they make much less insulin. And again, in response to a blood glucose challenge, they do not make as much insulin as their non-perturbed counterparts. So what this really showed us was that this postnatal day 12 to postnatal day 21 window is really important for the microbiota to instruct what's going on in the pancreas with respect to beta cell development. So we wanted to then go and ask whether or not there is this kind of temporal window in humans as well. Of course, we can't go around and antibiotic treat a bunch of kids like we are able to antibiotic treat mice. So the way we approached this question was to obtain infant stool samples. So we obtained stool samples from babies that were less than 6 months old, so 0 to 6 months old, 6 to 12 months old, 12 to 18 months old, or greater than 18 months old. And then we would take those stool samples and we would colonize mothers And then those mothers would give birth to pups, and we would analyze those pups for beta cell mass at P20. So those mothers would pass on that microbiota to those pups. And then we would look at beta cell mass. So when we do this experiment, what we found is that this red line indicates what the beta cell mass looks like in just a germ-free animal. So if you colonize animals with infant stool from 0 to 6 months or greater than 18 months, despite the fact that a lot of these microbes colonize these mice, it really doesn't have a huge impact on the induction of beta cell development in these mice. But what was really intriguing to us is that when we put in the microbiota from a 6 to 12-month-old child from multiple infants, we find that this microbiota potently induces beta cell mass in mice. Now, what's also interesting about this window of life, if you remember, I also pointed out that the 6 to 12 month period of life is also the same time when human infants are undergoing the most beta cell proliferation. So this suggests that what we're finding in mice, that is that there's a temporal window of life where the microbiota is really instructing beta cell mass, is also true in humans. So the question becomes, who are the microbes that are really driving this response and really relevant during this early window of life? And we did a lot of different sequencing experiments that involved single antibiotic perturbation. We also did many experiments with probing the fungal response. I don't have time to get into that data, but we did identify that fungi might be relevant based off of some of these studies. And since I had just shown you that antibiotic perturbation resulted in a depletion of beta cell mass, I also wanted to share with you what happens when you deplete fungal communities. And these are communities of the microbiota that we don't often think about. So if we do the same experiment with fluconazole, which is an antifungal, during these 3 windows of life, again, what we find is that this particular third window of life is most important, that when you deplete with fluconazole, we see a significant reduction in beta cell mass in adult animals that had their fungal communities perturbed. Also, interestingly, when you go and look at the colonization of fungi, we see very dynamic fluctuation of fungal communities during this early life window, where we see quite a few fungi that are colonizing the gut in the opening of this window around P12. And then we see a dramatic drop at the end of this window of colonization of fungi, most of which are in the Candida genre. So it seems like these early life fungal communities are fluctuating a lot, but certainly relevant to beta cell development, as are bacterial species. So based off of all the sequencing experiments that we did, we isolated organisms from neonatal mice or neonatal human infant stool, or we purchased organisms that we were unable to grow that were relevant in our sequencing experiments. We then used these organisms singly to colonize germ-free animals, and we wanted to ask who's sufficient to induce this beta cell development. And of the organisms that we chose a neonatal E. coli isolate, an Enterococcus species, a lactobacillus species, and then 2 fungal species that we know colonize the human infant gut, Candida dubliniensis and Candida albicans. So again, to orient you to beta cell mass in a germ-free animal, significantly less than when compared to an SPF animal. Now, when you take any of these single bacterial organisms, including this neonatal E. coli isolate and this Candida dubliniensis, we see that these 2 organisms most robustly induce beta cell development in these animals, as well as this Enterococcus species. However, not all organisms are sufficient to do this. In fact, very closely related to Candida dubliniensis is Candida albicans, is not able to induce beta cell mass in these animals, nor is a very common neonatal bacterial organism, lactobacillus. So there are multiple organisms that are capable of doing this, but not any organism will do. So we had identified a handful of organisms that were sufficient to induce beta cell mass in our animals. So we wanted to understand, well, what is the mechanism by which these organisms can do this? So Jennifer Hill had published another really amazing study in Karen Gilliman's group where she had identified a mechanism in zebrafish by which a bacteria could produce a molecule which they termed BEF-A that could poke holes in beta cells and cause them to proliferate. So they had identified a direct effect by which their microbes in zebrafish were able to influence beta cells. So we had tried for quite some time to identify secreted metabolites that any of our organisms could have a direct effect and failed in all of those attempts. So it seemed that our microbes were using a more indirect mechanism to influence beta cells. So we started looking at immune-mediated mechanisms, and this really came from an analysis, an unbiased RNA-seq approach, where we looked at islets isolated from either germ-free or SPF animals. And when we looked at the major transcripts that were lost in germ-free animals, transcripts that were associated with macrophages were really depleted in mice that did not possess a microbiota. And macrophages are the major immune cell type present in the pancreas of a healthy animal. You don't see a ton of T or B cells. It's really macrophages that are the relevant immune cell type. So we wanted to ask the question of whether or not macrophages played any role early in life in beta cell development. So we used a variety of ways to do this. I'm just going to share with you one way that we approach this. And we use clodronate, which is a liposome-mediated way to deplete macrophages in mice. And we depleted these macrophages again in animals. These are just SPF animals, and they're treated with liposomes between this window of life, P7 and P20. We widened the window a little bit here. And we know that we can rapidly deplete these macrophages with these liposome injections. Once we stop the liposome injections, macrophages do rebound in these mice. And then we analyze these animals at adulthood. So really asking the question, is it early life macrophages that are important for beta cell establishment? So when she did these experiments, indeed, she found that when you deplete macrophages during the same window of life, that you see a decrease in the insulin producing capacity in animals and significantly reduced serum insulin levels. And consistent with this, animals that had early life depletion of macrophages, these animals are unable to clear glucose as well as their unperturbed counterparts. So this is really demonstrating that indeed macrophages during this early life are important for helping to establish lifelong beta cell development. So the question to us became, is it the bacteria? Are they functioning through the macrophages in order to have the effect on the beta cells? So the first question we asked was, are any of these organisms that we found to promote beta cell mass, do they have effect on macrophage seeding of the pancreas? So the first thing we looked at was, is the microbiota relevant in seeding the pancreas with these macrophages? And indeed, if you look at germ-free animals, there's significantly less macrophages in each individual islet. And if you colonize these animals with various organisms that we know are beta cell promoting, we find that these organisms indeed also promote macrophage induction in these islets. What was interesting in that is that most of the organisms we used, including non-beta cell promoting organisms, seem to be good at inducing beta cell mass. But Candida dubliniensis seemed to do it the best. So in order to ask whether or not the ability of these specific organisms, in particular Candida dubliniensis and Enterococcus, were functioning through macrophages, we did a study where we monoassociated animals with either Candida dubliniensis or Enterococcus, and then in half of those mice, we would also deplete the macrophages to ask whether or not the effect on these beta cells was macrophage dependent. And while we did not see a difference in Enterococcus colonized animals when we depleted macrophages, Enterococcus was able to do this with or without macrophages, suggesting that Enterococcus does this through a distinct mechanism. when you colonize animals with Candida dubliniensis and deplete those macrophages, now you lose Candida dubliniensis's beta cell promoting effect. So the last kind of mechanistic piece to this story is, check my time here, is understanding how is it that Candida dubliniensis is able to promote beta cells or promote macrophage seeding in the pancreas, whereas other organisms cannot. We really want to try and understand this specificity. So we were able to obtain multiple strains of Candida dubliniensis. So the one strain that we're using that promotes beta cells really well in mice is a CD36 strain. Another strain that we were able to obtain is called a Wu284 strain. These are both strains that are commonly used in the field, but have very disparate effects when it comes to beta cell mass, in that this Wu284 strain does not promote beta cells in these animals. These animals look very much germ-free. However, when we obtain strains of this non-beta cell promoting strain that are either, that are either lacking enzymes that promote mannose, or strains that promote hyphal formation, what we find is that hyphal formation in this organism doesn't seem to play a role in its ability to promote beta cell mass. But when you modulate the cell surface or the mannose that's in the cell wall of these fungi, this strain that didn't produce beta cell mass before now is able to. So we're trying to understand the nature of this. In our paper, we published that the cell wall is very different in these strains, and there's a lot less mannose in the beta cell promoting strain. So we thought that it was going to be something in the cell wall, but since we have identified that it's something that the fungi is actually secreting that is responsible, so that secretome is very different in these different strains. So just to summarize, I'm going to end by talking a little bit about how we think about these things therapeutically. But we've identified that there are specific organisms within the neonatal gut. In particular, we focus on neonatal fungi. And we've identified that they secrete something that influences macrophages that then help to promote appropriate beta cell and insulin producing capacity in these animals. So we're really trying to identify, of course, mechanistically, what is that molecule? But we really wanted to understand whether or not we can harness this therapeutically, because I started off by talking about if we can understand things that influence beta cell development, we might be able to reverse or treat type 1 diabetes. So we approached this really in two ways. And these are my last two slides. The first one was, if you're colonized with a beta cell promoting organism, so Candida dubliniensis, or if you're colonized with an organism that does not promote beta cells, does it change your trajectory for developing type 1 diabetes? And the model that we use for type 1 diabetes is germ-free non-obese diabetic mice, or the NOD mouse model. And these animals spontaneously develop diabetes over a long period of time. So we colonize these germ-free mice with either Candida dubliniensis or lactobacillus, and then we watch these animals develop diabetes. And what we found is that animals that were colonized early in life with a beta cell promoting organism developed a lot less diabetes. In fact, only 30% of the colony developed diabetes, whereas animals that were colonized with a non-beta cell promoting organism, up to 80% of those animals in that colony would develop type 1 diabetes. So suggesting that perhaps having the right organisms can prevent or promote you from developing type 1 diabetes. But can we harness this therapeutically in adults? It's one thing to be able to do this in an infant, but what about individuals who are already adults who already have type 1 diabetes? Can we use this organism to affect adult beta cells? So to test this, we took germ-free animals, and you're able to deplete beta cells in these adult germ-free animals with a drug called streptozotocin. And it readily depletes beta cells. So we depleted all beta cells in germ-free animals. And then we separated out these animals and we treated them orally with either Candida dubliniensis, or we left them germ-free. And when we did these, we found pretty striking results in that animals that were left germ-free, we lost about 70% of those animals, whereas all animals that were orally treated with Candida dubliniensis survived this process. Of the surviving animals, we found that the ones that were treated with Candida dubliniensis could clear blood glucose, and their beta cell mass was significantly increased when compared to animals that were germ-free. So this is really the first suggestion that perhaps some of these neonatal signals can be harnessed in adulthood to promote beta cell mass. So I just wanted to end with this idea that these transient disruptions, so a lot of times we're looking at adults who have a disease and who do not have a disease, but we have no idea what had happened to them during their early life. But these transient disruptions to a developing microbiota can really have these lifelong consequences. And understanding this interaction, I think, is going to be important to a lot of different diseases. And again, driving home this idea that missing certain microbes, there is intimate relationship between these early life microbes and the host. And lacking some of these signals can really be the reason that we go on to develop disease later in life. So I'll end there, and I've already acknowledged the collaborators in this project, but I also like to highlight some really important individuals on my lab. Stack Stevens already talked about, but also Ricky Bell, who's my lab manager and gnotobiotic facility manager. We can't do any of these technical germ-free experiments without her. And I also want to just highlight a technician that's been working with our groups for over a decade. Unfortunately, she just lost her battle with cancer, and we really miss her. We lost her last week. So I just want to acknowledge her. So with that, I will stop. Thank you.
Thank you so much, Dr. Ron. That was an amazing talk. I'm sorry for your loss.
Thank
And
you.
also, yeah, it's amazing to see just like another example of how the microbiome and its development in early life play such a pivotal role in our development of disease. I think
Yeah,
we have a time for maybe a couple questions.
sounds great.
Um, so first question, as they ask or they say, it's interesting that bacteria and fungi appear to have a critical window of colonization. And they ask, do you have any indications of other members, like looking at other orders of life, like back to phages or maybe archaea, and maybe the potential for them to have effects?
I love that question because I have a large part of my group who's currently working on bacteriophages. We have not addressed that with respect to this beta cell mass phenotype, but it's something that is on our minds to do. And then we just don't work with any archaeal species, so I don't know. But I love the question.
Also, yeah, I think just it's nice to see you highlighting like the role of fungi. I feel like so often we really zoom in on the bacteria and think they're the only thing that matters, but
Mhm.
it's nice to see that there's other guys out there too that need to be considered as well.
Sure.
And then we have maybe one other question. So they ask, did you see whether and how much basically, they're asking if there's any synergy between the bacteria and the fungi. Like, did you look at the effect of maybe having them together?
Um, we are doing those studies now because it's a really good question to ask if there is synergy or bacterial, because fungi and bacteria definitely interact in environments, and this has been shown so elegantly in lots of soil studies. I don't have any results yet, but we are certainly asking those questions if there's any synergy between them.
Yeah, thank you again so much, Dr. Round, for speaking today. And I think maybe that's all the time we have for questions.
Great, thanks so much.